Now we'll study the different types of fermentation processes. The major fermentation products that are produced by commercial important fermentation processes. These products, fermentation products, which are produced on large scale, are listed as microbial cells. That means they are produced in the form of biomass, for example, biofertilizers, biopesticides, yeast, etc. Second, microbial enzymes, which are produced by using fermentation methods from utilizing bacteria and fungi. Then, different microbial metabolites that includes different types of organic acids like citric acid, lactic acid, acetic acid, different types of vitamins like vitamin B1, vitamin B12, etc. Different types of amino acids produced by bacteria and fungi like glutamine, lysine. Several types of antibiotics produced by majority fungi and actinomycetes, mainly penicillin and streptomycin, and many more. Different types of solvents like acetone, butanol. Fermentations are carried out in order to produce different recombinant products. It includes interferons, insulin, human serum albumin, factor 8, 9, epidermal growth factors, calf chymosin, bovine stomatostatin, and different types of vaccines. So all these are produced as a result of recombinant products. These are produced from recombinant microbes. Then there are different types of animal cell products that includes immune sera. So we'll be discussing about typical examples of all these different categories of fermentation products here on. The products which are produced by microbial transformation process such as steroids and vinegar will be the additional technique apart from fermentation process that will add to our knowledge, as it is not a conventional fermentation process. It is a different type of slightly different type of process that is called as transformation. Now first we'll try and understand how many different types of fermentation processes are there. These are the types of fermentation processes which are based on what is the method of production, what is the nature of raw material. So first in that is basically fermentation processes in general. We can divide them as submerged fermentation process and solid state fermentation process. Let us see what is this submerged fermentation process. In basic, submerged fermentation process makes use of liquid medium. So liquid medium is used for the production of microbial metabolites. So the medium, the nature of the medium which is used in submerged fermentation is liquid. Liquid means presence of excess water. So this is used in case of deep tank fermenters. So there is use of deep tank fermenters with the facility of mixing, aeration, bubbling of air, temperature control, pH control, sampling facilities, and other automatic control systems. A typical design of fermenter we have already discussed. Now, the major features of submerged fermentation are these fermentations are carried out in presence of excess water. So ample of water is used as a solvent for making the fermentation medium. So this is a very important characteristic. The advantage of this submerged fermentation is the monitoring and operation and control of this process is automatic and it is easy. There are different sizes of fermenters which are available for the production, which we have already discussed in the different levels of fermentations under the scale up topic. We already discussed this. 2 to 10 liters for laboratory fermenter, this is the capacity. 25 to 100 gallons for pilot plant fermenters, and 5,000 to 10,000 gallons or 250,000 to 500,000 gallons for large scale production, large fermenters. Therefore, the equipment cost, that is non-recurring, the capital cost in this submerged fermentation is very high. But the sampling is easier, and due to automatic control, there is saving of the cost of labor. These submerged fermentation processes, it can be further carried out, or we can say they can be divided into different subcategories. Submerged fermentation, it can be carried out as a batch fermentation, continuous fermentation. There is one more in between, that is semi-continuous fermentation and feed batch process. These are the three categories of submerged fermentation. So submerged fermentation using excess water, it can be carried out by batch way, continuous way, or feed batch way. Let us understand what are these three categories. Batch fermentation, as the name indicates, batch. This implies to the limit of fermentation medium. So it is a closed culture system which is carried out in a fermenter. The process is carried out in a fermenter which contains an initial and limited amount of nutrients. All these nutrients are prepared in water. So this is very important feature of batch fermentation, that it contains limited amount of nutrients. The inoculation culture will pass through the inoculated culture will pass through a number of phases which we already learned, such as lag phase, log phase, or stationary phase, logarithmic phase, that is exponential phase, stationary phase, and death phase. That means the whole life cycle of the bacterial cell, it occurs in this closed culture system due to limitation of nutrients. It is called as batch fermentation. Second is continuous fermentation. In continuous fermentation, the exponential growth in batch culture, it is extended or it is prolonged. The growth phase, exponential growth phase is prolonged. That is the log phase is prolonged. And how it is prolonged? It is prolonged by the addition of fresh medium to the vessel. So there is continuous addition of fresh nutrient medium to the fermentation vessel. This will cause the increase in the volume of fermentation broth in the vessel. Therefore, at the same time, whatever the excess amount of liquid is added, that is called as the overflow. So there is an overflow device which is fitted to the fermenter to adjust the rate of addition of fresh medium equal to the rate of loss of culture. This means that in case of continuous fermentation, there is continuous addition of nutrients from one end, and there is continuous removal of the culture broth to maintain the appropriate size or volume of the medium in the fermenter. So if the medium is fed continuously to such culture at a suitable rate, a steady state is achieved eventually, that is formation of new biomass by the culture is balanced by the loss of culture from the vessel. So the rate of formation of new cells is balanced by the rate of loss of cells from the culture, and that leads to the presence of a steady state. That means most of the cells present in fermenter, they will occur or they will remain in the same growth phase, that is exponential growth phase, that is called as continuous fermentation. The next process is fed batch fermentation or fed batch process. As the name indicates, it is a variation of batch process. So we already learned what is the meaning of batch process, and the major feature of batch fermentation is limitation of the volume of medium. Now this is a slight variation in batch process. So it is a batch culture. However, it is fed continuously. That means fed means there is continuous addition of fresh medium, or it is sequentially added with medium. But how it is different from this continuous fermentation, that there is no removal of culture fluid. So there is addition of fresh medium without the removal of culture medium, which happens in case of continuous fermentation. So this is the flow chart that shows the different categories of fermentations. In case of submerge, this is the batch 1, as the diagram indicates, fermenter with impeller, agitation, automatic control facilities, and this is the volume of medium which remains constant. So after the inoculation, the fermenter is operated for definite period of time, and after the incubation period is over, the broth is ready for harvesting. This is a very commonly used method for the production of different types of microbial metabolites. There is a relatively low substrate utilization rate, and there is low risk of contamination and strain mutation. Second category is fed batch. So as we can see that this arrow is shown here, that there is addition of fresh nutrient medium, but that medium is also added in limit till the proper volume of this fermentation medium is achieved, because there is no outlet, there is only inlet. So it is best during substrate inhibition and catabolite reparation, prolonged log and stationary phase. That means the objective which is achieved in continuous fermentation is also achieved in fed batch culture. That means we can bring the culture into log phase and stationary phase for a particular period of time. Fed batch process. The effectiveness over batch is due to concentrated substrate utilization. So we can add the substrates in the form of concentrated form, large metabolites production during stationary phase. So this system seems to be good for the production of microbial metabolites which are produced at the end of log phase or in stationary phase. The third category is continuous fermentation. So we can see here in continuous fermentation there is an inlet which indicates there is continuous addition of fresh medium, and there is outlet which indicates the loss of culture. So this continuous fermentation has less sterilization and re-inoculation. This is the uh, advantage. So frequent sterilization is not required and re-inoculation is not required. That saves the power and that saves the culture as well. Less maintenance cost and faster substrate utilization rate. These are the advantages. Effectiveness of continuous over others is due to high productivity and it reduces uh, butanol inhibition in case of typical fermentation. This is a fed batch fermentation process. We can see here this is the fermenter and this is at the volume of the medium. Initially it is less, then as the fed batch fermentation process starts, after some time there is addition of fresh medium to this fermenter and the volume then rises. Again, a particular time is allowed for the microorganisms to grow and to produce the microbial metabolites, and again after some time the fresh fermentation medium is added and again the volume rises. So this is continued till it rises to a particular level which is already decided. So once it reaches to that particular level, the addition is stopped. The incubation, after the completion of the incubation period, allotted incubation period, the fermentation is stopped and the broth is harvested. That is in case of fed batch process. This is again a simple diagram that shows a basic difference between batch fermentation, continuous and fed batch process. We can see in batch there is a limitation of volume of the medium. In continuous there is addition of fresh medium and there is removal of culture. So addition and removal, that is a feature of continuous. In case of fed batch there is one way addition till the volume of fermentation medium is achieved to the desirable one. There is no outlet. So these are the differences between batch, continuous and fed batch fermentation. Second type of fermentation category is solid state fermentation, or it is also called as SSF. In many books you will find that it is also called as solid substrate cultivation. Solid substrate cultivation, that is SSC. Now the key feature of this solid state fermentation process is this process it occurs in absence or near absence of free water. So that is the difference between submerged and solid state. In case of solid state we can note that there is use of excess water, and here there is absence or near absence of free water. That is a major difference. And another key feature you can see that it is solid substrate. So the nature of raw material, we can see that the nature is solid. So the substrate which are used in solid state fermentation, they are utilized as the raw materials, and this raw material, it supplies the majority carbon source, nitrogen source, and other essential nutrients which are required to carry out the production of desirable metabolite by inoculated producer organism. So this raw material, it supplies all the essential nutrients. It provides physical support. This is very important. So it serves as a physical support as well as it serves as a source of nutrients. There are several natural materials which are used as a substrate to be utilized in solid state fermentation. These are the several examples of natural materials, and all these natural materials are the agro waste materials which are obtained from different agro based industries. It is a type of waste, and this biological waste, it can be reutilized in solid state fermentation as substrate. These examples are sugarcane bagasse. So this sugarcane bagasse, it is the waste which is obtained or that remains after the extraction of sugar from sugar cane. So whatever things that is left behind, it is called as sugarcane bagasse, which is very useful to use as a physical support during solid state fermentation. Sawdust, this is the remains of the processing of wood. So this sawdust, it can be also used as a physical support in which there can be uh, impregnation of nutrients. Tea waste, tea waste, it is obtained from tea processing industry. So after the removal of desirable tea, processing of tea, whatever the waste that remains, so it can be used as a substrate. There are different types of fruit pulps which are obtained from food processing industries. For example, apple pomace. This is apple pomace म्हणजे मराठीमध्ये चोथा. So this apple pomace, it can be, it is still nutritionally rich and it can be used as a substrate. Then there are different types of oil processing industries, and these oil processing industries generate oil cakes as the major waste. Oil cakes, so it is, oil cake is the waste that remains after the extraction of oil from different types of seeds and different veg materials like coconut oil. So coconut oil cake, palm kernel cake, soybean cake, groundnut oil cake, pomegranate cake. So likewise, these types of oil cakes that remains or it is obtained from the oil processing industry that can be used as a substrate. Then wheat bran, which remains, which is the byproduct which is obtained from the processing of wheat, which is rich in cellulose. This is cassava. Cassava, which is similar to, it is a type of tuber which is grown in South India, which is generally similar to, मराठीमध्ये त्याला रताळ म्हणतात. So this is a tuber or root. So this cassava, after, which is rich in carbohydrates, so after the carbohydrate extraction or after the processing of these tubers, whatever the remains which are left behind, that can be used. So cassava, the waste which is obtained from cassava, it can be used as a substrate. Then this is the barley. So we know that this barley is used in the making of alcoholic beverages, for example beer, and whatever the remains which are thrown away from that industry, it can be used for animal feed, as well as it can be used as a substrate for solid state fermentation. Then this is corn corn cobs which are left behind after the removal of corn grains. So it can be a good material for uh, to act as a substrate in solid state fermentation. This is coffee husk. That means husk means this is a tough covering over the coffee balls, and after the processing of this coffee, whatever the waste that is generated, it is called as coffee husk. So this husk material, it can be utilized as a substrate for solid state fermentation. Likewise, the waste which is obtained from the processing of tea, which is called as tea waste. And of course, this is barley. So after the mashing process, during the making of beer, these barley grains are ground, milled, then they are processed. The process is called as mashing, and after mashing, uh, the water part is separated, and that is used for the fermentation of or the making of beer. But whatever the insoluble material that are left behind, that is म्हणजे हा चौथा असतो बारलीचा. So whatever the insoluble material that remains, that is called as spent barley material. So spent barley material, it is the waste which is obtained from beer making industry, which can be used for animal feed, as well as it can be used as a substrate for solid state fermentation. So these are the different types of substrates which are used in the solid state fermentation. So what are the equipments which are used for solid state fermentation? So these fermentations are carried out using trays, beans, and rotating drums. Majoritarily, there can be use of different materials, and there are different methods of solid state fermentation. These are the common materials: trays, bins, and rotating drums. Now, in solid state fermentation, the selection of microorganism is going to be a very key factor, because microorganisms differ in their ability of production of microbial metabolites by different methods. We'll find that certain bacteria and fungi, the production of microbial metabolite is higher using submerged fermentation, and the production is higher sometimes using solid state fermentation. So identification of the characteristic of that particular microorganism is very important during the selection. So therefore selection of microorganism is a key factor in order to get improved yield. So SSF, it can be used for the production of microbial metabolites by bacteria as well as by fungi, actinomyces, and yeast. The optimization of this process, it is based on common features which are used for the scale up or optimization of submerged fermentation, like inoculum size, initial moisture, pH, pre-treatment of raw material. It is frequently required because it is a crude raw material, and therefore it may require pre-treatment. Then relative humidity and maintenance of moisture is very important, because there is absence of free water. Then there is a provision for aeration and agitation, but it is different than submerged fermentation. Optimization is done on the basis of requirement of nutrients such as nitrogen, carbon, phosphorus, and trace elements, inducers in case of enzyme production, extraction, and purification methods. So this solid state fermentation, it is very important in order to get very higher yields in case of different types of products, such as this process is good for production of microbial enzymes like cellulase, protease, pectinase, xylanase, amylase, lipase, etc. etc. There are examples of production of organic acids and bioinsecticides like Trichoderma Bacillus thuringiensis using SSF. It is also useful in the making of different animal feeds, including production of yeast and algae, that is single cell protein. These are the different examples of the processes and the products. Koji fermentation process, then Agaricus pleurotus, shiitake, mushrooms are produced using SSF. Then sugarcane bagasse and pulp, it is good for making of compost. So making of compost, it is also a solid state fermentation. Composting is a solid state fermentation. Then production of different types of biocontrol agents like production of biomass like Breweria, Trichoderma, production of products like plant growth promoting products like gibberellins, then microbial products, biomass like Rhizobium, Trichoderma. Production of different types of enzymes is also done by using solid state fermentation, that includes amylases, cellulases, proteases, pectinases, xylanases. Antibiotics are also produced like penicillin. Different types of probiotics are also produced. There are examples of production of citric acid, fumaric acid by using SSF, alkaloids. Then in case of ethanol production also during malting and brewing, SSF is also technology is used. So basically, what is this solid state fermentation technology? There is use of solid substrates which has sufficient moisture or water content, but there is no free water present, and microorganisms are inoculated into this solid material. Microbes are mixed, and then it is exposed to desirable temperature for desirable incubation period, and the whole material is then used for extraction and purification of fermentation products. This is a typical design where we can see that there is a incubation tunnel, and these are the trays, and these trays are spreaded with culture and substrate. So this substrate and culture, it is mixed and it is spreaded into the trays in thin layers or whatever the thickness of the layer which is previously decided. This cultivation tunnel is having facilities like there is provision of cooling devices, and there is a provision of supply of oxygen. There is provision of an outlet that removes the hot air, and so this is the provision for the maintenance of incubation temperature, because this process seems to generate lot of energy, and energy is released in the form of heat. So a lot of exothermic reactions may happen during this solid state fermentation due to the growth of microorganisms, And in order to control the temperature, there has to be an outlet and presence of cooling system, that is through the passage of cool air, or it can be this system, it can be jacketed. There can be a jacket through which cooling water it can be passed in order to maintain the temperature. So this is the tray system. Yeah, so this is called as a bioreactor, because a reactor is what? It is a system in which the reactions are going on with the help of microorganisms, and therefore we call it as bioreactor. So we call this as a tray bioreactor. We can see this is a cultivation tunnel, and these are the trays. These trays are spreaded with substrate and microorganisms. Of course, this substrate is sterilized. The trays are sterilized, so they are made free from microorganisms first, and then this sterile substrate is mixed, or it is inoculated with desirable inoculum or microorganism, and there is provision of supply of air and temperature maintenance, and therefore it is called as an incubator. So this is a tray bioreactor. Like this, you can see that this is a big tunnel, this is the lead, and the person is placing the trays inside this cultivation tunnel. This is a big system that you can see. Mostly we can see this in case of production of yeast, production of maturation of cheese, and production of mushrooms. So you can see these are the stacks of trays in which the substrate is mixed with the microbe, and this whole room has a uniform temperature. So this whole is a cultivation tunnel which has facility piping network for the control of temperature and moisture. This is the another reactor in solid state fermentation. It is called as packed bed reactor. So the difference is, you can see the thickness of this substrate, the thickness of this bed layer is more in case of packed bed reactors as compared to the previous technique of tray bioreactor. So that is the difference. This is again a cultivation tunnel which has provision of passage of air and removal of hot air. The substrate and microorganisms are supported here. So this is a thick bed called as packed bed bioreactor. Then this is very interesting technology. This is generally useful in the composting process mostly. So it is a drum bioreactor. It is a drum bioreactor. So the mechanical design is like this. There is use of a drum. So these drums, they can be of rotating type, or these can be of stirred type. Now rotating drum bioreactor, what is it? This rotating drum bioreactor, we can see that this is the substrate which is inoculated with microorganism, which is loose and free enough to move the material. It can be moved, it is not very hard, and this drum is rotating in this fashion, and this provides the entry of air inside this substrate, for that facilitates the aeration. So there is a inlet outlet. This is again a temperature controlled drum. Second, so the difference is in relation to the mixing facility. In case of second one, that is stirred drum bioreactor, this stirred drum bioreactor has a similar mechanical design. Only the difference is that it has a, an impeller which has a function of mixing. So this is continuously rotating, and this is responsible for the mixing of this substrate that facilitates aeration and that facilitates the heat transfer, which is very important for the distribution of heat or distribution of to maintain the uniform temperature among this whole substrate. So that is another design of drum bioreactor in solid state fermentation. This is again a design of drum solid state fermenter. This is the drum which is equipped with several facilities. These are the major differences between solid state fermentation and submerged fermentation. So I'll just quickly go through this. Low water contained, high water contained, uh, three-phase system. It contains gas, liquid and solid. This is a two-phase system, gas and liquid only. So solid component is missing here. A complex substrate insoluble in water, high local concentration of nutrients. Here nutrients are dissolved in water, and therefore the concentration is less. Here there is less water, therefore concentration of nutrients is higher. Non-homologous system, this is homologous system. Microorganisms are grown on surface of solid substrate. Here microorganisms are grown inside the liquid medium. In case of SSF, gas, liquid and liquid solid oxygen transfer. In case of submerged, it is gas, liquid, oxygen transfer only. SSF has limitations in heat, oxygen and nutrient transfer, but that is not the problem in case of submerged fermentation. The exception is can be oxygen transfer. In case of SSF, the heat is removed from a stream of air by replacing bioreactor into temperature controlled temperature. In case of submerged, there is a use of cooling jackets. SSF, the process monitoring and control is sometimes difficult, but in case of submerged process, the online monitoring and control system makes the process easier. In case of SSF, high concentration of the product is obtained. In submerged fermentation, product is dissolved in liquid phase. So, and therefore there will be differences between the downstream processes of solid substrate fermentation and submerged cultivation process in the initial steps.